He has a book out called Connectome, The Brain's Wiring, How the Brain's Wiring Makes Us Who We Are. And at MIT, you can't just be a professor in one thing. He's a professor of computational neuroscience and a professor of physics. Please could we welcome Sebastian Seung.
Those are fantastic talks. That really makes me wish I was part of the Media Lab. Alas, I'm not. I'm in the neuroscience department at MIT. My name is Sebastian, and this book is about what I believe is the importance of mapping out the brain's connections for understanding how the brain works and why it malfunctions. And the short pitch in my talk, I want to persuade you that you can all be neuroscientists because my lab has created a website where we're inviting the world to volunteer, help us analyze data, and help us map out the brain's connections. But before I get to that message, I'd like to say a little bit about how I got to be where I am today. So as a kid, I was at least a little weird. And so instead of football players and baseball players, I had other kinds of heroes, mathematicians and scientists. And a great mathematician was Pierre-Simon Laplace. And I read a story, an anecdote, maybe apocryphal, about how one day Laplace and Napoleon had a conversation. Napoleon said, "They tell me you have written this large book on the system of the universe and have never even mentioned its creator." And Laplace answered, "I had no need of that hypothesis." Actually, how many people here have heard this story? If you heard the story, raise your hand. Actually, not that many, I guess. So, the background is that Laplace had written a whole five-volume treatise on the motions of the planets called Celestial Mechanics, right? The motions of the planets around the sun. And he applied Newton's law of gravity. And he very precisely showed that he could explain with incredible accuracy the motions of the planets around the sun. So when I think about this, I wish I'd been a fly on the wall, because I want to know a little bit more about this background of this story. Were they at a dinner party? Were they in Napoleon's office? And exactly how did Laplace say this? Did he say it modestly? Like, "Oh, I had no need of that hypothesis." To be modest before Napoleon, and maybe even more importantly, before God? Or was it more triumphant? Like, "I'm a badass mathematician. I had no need for that hypothesis." He had a right to be triumphant because Napoleon had merely conquered Egypt, and Laplace had just conquered the universe. I'm not sure, but that story was part of what inspired me to study physics in college and in graduate school. But in graduate school, I actually grew disenchanted with physics, and I resolved to study the brain. What was so attractive about the brain? Well, in the case of physics, people are pretty convinced, at least in the everyday world, we have no need for the hypothesis of divine intervention to explain how things work in our everyday lives. Maybe if we want to talk about the beginning of the universe, the end of the universe, the very small, the very large, then science fails us. But in the case of the mind, it's not as clear. In fact, the mind seems the most challenging to explain on the basis of physical laws. And in fact, yesterday we heard from Kumare, my favorite false prophet. I loved his talk. And he asked people in this room, how many of you were religious? And I was shocked by how few people raised their hands. And so I would ask another question, which is, how many of you believe in a soul? If you believe in a soul, raise your hand. A little more, but actually not that many. But most people I talk with, if you get into conversations with them and they hear you're a neuroscientist, they're really excited, they're really interested, they want to hear about how the brain works. But in the end, the conversation kind of comes around to the fact that they believe that you can't really explain how the mind works without invoking something supernatural, without invoking the soul. And that belief, of course, I think that's why neuroscience has a certain glamour to it, why it's fashionable beyond all proportion to its accomplishments, because of that mystery. And in fact, this objection goes back a long time. Here's another, in those days they had these, you're not only known for your intellect, you're known for your hair. But Leibniz, he wrote this a long time ago. He said, in imagining a machine whose construction would enable it to have perception, one could conceive it enlarged so that one could enter into it, just like into a windmill. Supposing this, one should find only parts pushing on one another, and never anything by which to explain a perception. So Leibniz believed that no machine could perceive. And this really is, in the end, the big question. Can we think of the brain, can we understand the mind as the workings of a very complicated and marvelous machine, but still something that's a machine? It's not clear. I would like to say, neuroscientists would want to say, I have no need for that hypothesis. We have no need to invoke something else. But we can't say it triumphantly, definitely, because we haven't really gotten there. We haven't really explained how the mind works or how the brain works. And people don't necessarily understand that. They think, oh, you neuroscientists, you know so much. But they say, fix my brain. There's all these mental disorders. Can't you fix it? And so this is not a philosophical question merely, just as is the brain a machine. I think these people who are asking for the fix, who are impatient for the fix, they believe the brain is a machine, and therefore it ought to be possible to fix it. But even if the brain is a machine, it's a machine that we yet have almost no understanding of. That's why it's so hard for neuroscience to make progress in solving the problems we all care about. And that's why most of my work, I do a little work that really is oriented towards disease, but most of my work is really oriented towards understanding how the brain works normally, which is a miracle enough by itself. It's not surprising that it malfunctions, to me. What's crazy is that it works. Okay, so what progress are we making towards understanding perception? I would say that neuroscience is making progress in baby steps because we have new technologies. The brain is a complicated machine, and therefore we can't expect to understand it without having equally or maybe even superior technologies to take it apart. And so let's start with an organ of the starting point, really, of visual perception, the one that we heard about from Ramesh, the eye. What's inside an eye? Has anyone here ever dissected an eye? Yeah? I highly recommend it. It's not as gruesome as it sounds. Just the next time you're cooking a fish, there's that head. Just take the eye out and slice it open. See what's inside. Here's one thing you'll find. This is the lens from a cow eye. It's a beautiful optical instrument. As you can see, it has this magnifying power. And it's kind of shocking that inside the eye, there exists something which seems like it was designed by someone or some entity that knows the laws of physics. And that's why William Paley, Charles Darwin, and Richard Dawkins over the ages have argued about whether we need to invoke a divine creator to explain the eye's existence. But actually, the lens is really the most trivial part of the eye. The most incredible part is this kind of thing that looks kind of like garbage, this pinkish thing here in the back, which is called the retina. But you can't see how incredibly marvelous this is without a microscope. And this first steps towards understanding the retina were taken by Santiago Ramon y Cajal. In 1900, he published a wonderful book called The Structure of the Mammalian Retina. And using a stain developed by an Italian colleague, Camillo Golgi, he was able to visualize for the first time the neurons which are inside the retina. What did that reveal? Well, at the top of this diagram on the right in his drawing are the photoreceptors. They're the cells that actually receive the light and convert it into electrical impulses. That's what you'd expect if the eye were like a camera. But there's more than those photoreceptors. There's all these other kinds of neurons, cells in the eye, and they're doing something. What are they doing? Well, it's such a complicated structure that it's safe to say that the eye is really more than just a camera. It's also a computer. It performs some of the tasks of visual perception. And so what I'm going to show you is some pictures of the retina, which are developed with a more powerful microscope than Cajal had. It's an electron microscope. It magnifies to a huge magnification. And we're going to look at a little piece of mouse retina, inside the little red square, which is just 1/3 of a millimeter across, we're going to look at an even smaller part of that picture. And so here it is right here. This is from the laboratory of Winfried Denk, and Kevin Brigman and Moritz Helmstedt are collaborators of mine. And so I'm going to just play this as a video. So what you're seeing is slice after slice of a tiny piece of the mouse retina. Each slice is 1,000 times thinner than a hair. At the tops and bottoms, you see the round parts of neurons. They're the cell bodies. They contain the nucleus, the DNA, and so on. And inside, between the two layers of cell bodies, is a whole tangled up spaghetti of neural branches. So those branches are intermingling, and those neurons are making synapses onto each other and talking to each other through those. And so if we want to find a single neuron in there, we have to trace out the paths of all the wires. Those branches of neurons are like the wires. They communicate through them. And so what we've done here is I've shown a rendition of taking one of the cell bodies at the bottom and tracing out all of its intricate branches, and we see the shape of a neuron. OK, so let me talk about a particular kind of neuron. This is called the J type of ganglion cell. And so you can recognize it because it's got a cell body, and it's got its branches pointing downwards. Most of the neurons in the retina have all their dendrites in all directions. This one can be recognized by the fact that it has them all downwards. And if we look at a side view of that, we see a wonderful kind of curved shape of the branches. And I found a nice picture of a tree, which looks almost like that. And of course, neurons are often compared with trees because of their intricate branches. What's known about the Jcell? Well, that's not just, we call it the Jcell because it's a type of cell. There's many cells of that type inside a retina. And here's a wonderful picture from the discoverers of the Jcell, a laboratory at Harvard run by Josh Sains. And they've genetically engineered a mouse so that every Jcell has become fluorescent. And so you can see that all of these cells have their dendrites, their branches, pointing downwards. So this is another one of the technologies besides imaging, which is the genetic control of the parts of the neuronal machine. Here's another kind of study which shows that, a property of the Jcell, which is that it responds to moving stimuli. So we all know that one of the most important functions of vision is to detect moving objects, right? So we can jump out of a moving car, hit a baseball, and so on. How is that done by the visual system? Well, this data here shows that. It looks a little complicated, but if you look around the circle, you'll see electrical recordings of this Jcell, and you'll see pulses, and each of those recordings corresponds to the response of the cell for stimuli moving in that direction. And so this cell produces a lot of pulses for stimuli that move downwards, and not for stimuli that move upwards. So it is able to discriminate between upward, downward, and sideways motion. All right, but the big mystery is we don't know how that develops. The photoreceptors are not direction selective, but the Jcells are direction selective. So how are we going to figure that out? So let me just show you a little bit about what that Jcell looks like in its habitat, because you might get the false impression that neurons are these islands surrounded by nothing. But in fact, there are thousands of neurons inside this retina. I'm just showing you a few of them. You saw enough to glimpse that there's a lot of wires inside there. And in fact, if I were to click on, if we had every neuron reconstructed inside that retina, it would be totally packed. There's a lot of empty space right now still, but it's completely, completely packed with those wires. And those wires are connected together, making those connections. And that's very important for the function of the network. OK, that was too complicated to look at. Here's another kind of cell in the retina. It has another beautiful shape, the Starburst amacrine cell. And so what we're analyzing these days in the lab is trying to figure out the neural network. How are all these cell types connected with each other? You can see the Jcell. You can see the Starburst cell in red. You can see a little red dot where there's a synaptic connection between them. And in green, there's another kind of cell called a bipolar cell. And all these cells are working together to enable the Jcell to detect motion. Okay, and so here's a kind of hypothesis that we're working on. One of the hypotheses for how this might work, it requires this circuit diagram, as it were. And just to give you an idea, you might ask, how many of those kinds of cells are in the retina? There's the Jcell, there's the Starburst cell. What's the parts list for the retina? And I just found this parts list, I think. This is probably the most parts list with which we are most familiar with and which frustrates us the most. And indeed, the parts list for the retina has tantalized neuroscientists for a century. Cahal worked on it. We're still not really able to, we still haven't succeeded in completely cataloging all this. Imagine that the eye is really like a jungle, and there's all these different kinds of tree, but we still haven't found them all yet. So here's a famous figure now from Dick Maslin showing about 50 different kinds of neuron in the retina. These days, people think it's more like 100 and still counting. So the retina, even just the retina, is an object of amazing complexity. How can we ever hope to understand it? And this is where you come in. So my lab has created a website called Eyewire, which is a small online community of amateur neuroscientists. And so any of you can go to that website and help us explore the retina. So that is a retinal neuron which is being reconstructed by our online community collectively. There's 6 users online right now. I can send them a little chat message. And on the right-hand side, you can see that here's a leaderboard with the national flags to the side. Usually this looks more international. Today it seems to be mostly Americans, but usually we have flags from all over the world. And so what's the game? Well, we've just zoomed in on a little chunk of the retina, a tiny little chunk. And you're seeing one slice through it, and we can navigate that slice up and down. And an artificial intelligence has colored in part of the branches of one of these retinal neurons, and your job is simply to find out where it made a mistake, because everyone knows that computers still aren't as smart as humans. So here you can actually correct the computer, and you can see that it should have kept on coloring right there. So we'll click. And we'll click again. And we'll click again. Oh, and now the AI finally got it and traced that branch all the way through this cube. So the first interesting thing that we found out when we put this online is that some people really like this. Now, maybe it's not surprising because everyone has enjoyed coloring at some point in their life. And this is like a really fancy coloring book because it's 3D. And the pages of the coloring book are images of the retina. And not only that, there's something kind of meditative about this. You know, for me, it kind of de-stresses. So after a hard day at work, I can sort of look at this. It's like watching the clouds. Some people say it's like a lava lamp. So there's a certain fun element to it, and it's fun to see your name on the leaderboard. You know, you can say unfinished. And there we are. Oh, I'm not on the leaderboard yet. I'll have to play a little more to get to be number one. Depends on how competitive I am today. All right, so that's the basic game. And I got this email from an Iwire recently, and he said, I must say how much I'm loving Eyewire. I never imagined there would be ways I could actually make a tiny contribution to real scientific discovery. It's a wonderful way to pass the hours, listening to science podcasts while I map neurons. Eyewire has become an important part of my life, and I feel a sense of responsibility for it, as well as personally gaining greatly from it. And sometimes my colleagues tell me, who are these people? And I said, you've lost your perspective. If they watched you in your lab every day, they would think you're crazy. Because science is an obsession. It is a certain kind of craziness. And through this website, we're able to share that craziness with other people. And people have fun. They found pieces of neurons that look like a little man in your retina. And there's always the goths. And I like this one. This is the person who said, I gave you the finger. So people reminded me that actually I went into science because it's fun. Of course, these days it's more like, oh, here are my colleagues griping about their something in the department, or I get my papers rejected, and I don't remember why I went into science, but then I see this kind of stuff and I remember, yeah, actually, it used to be fun. I should remember that feeling and try to keep on having it. And so November 26th, we're going to declare that J-Day. We want to kick off an Eyewire campaign to map out the connections between the Starburst American cells and the Jcells. And we want to answer this question about visual perception. How do Jcells detect downward motion? And I'm calling on you. I would like you to help us spread the word. Just to give a few minutes of your time, or your friends give us a few minutes of their time, we could discover something new in neuroscience. And I think this is just the beginning, because the brain is such a complicated object. Scientists can't do it alone. We need everyone. Professional scientists can't do it alone. We need amateur neuroscientists to help us. And I would say that there are these kinds of basic science questions, but also there are the questions that we all care about. Why don't our brains work properly? In some kinds of brain disorders, neurons degenerate and die, like Alzheimer's and Parkinson's. But there's plenty of psychiatric disorders for which no clear and consistent neuropathology exists. Neurons seem healthy. What's wrong? Well, for a long time, people have speculated. Maybe the neurons are wired differently. But as you can see from this kind of mess, how can you tell whether the wiring is wrong without actually tracing out the paths of the wires? That's the challenge we're up against. And I believe that if we all put our hearts and minds together, we could solve this problem. So please spread the word. J-Day. Remember that.
Thank you, Sebastian.